Hello, everyone. Welcome to our 27th edition of this INFOGEST Viva Webinar on Food Digestion. My name is Hernandez Olivas. So this time I'm actually introducing my colleagues, Raphaela and Marina. They're actually joining the organizing team. So welcome to this organizing activity. Well, for this activity. So this event is actually organized by a team of postdocs in Teagasc Research Center in Ireland. And this webinar aims to share the latest findings on the research for coming from postdocs, PhD students in the latest stage of her research, or young researcher overall. So the presentation this time in a pre-recorded video will be followed by a question and answer session with the speaker. So I actively invite you to use the button somewhere around that I think is in the bottom part of the screen. And then at the end of the presentation, we will just discuss this question. So maybe I can hand over Raphaela so she can introduce our speaker.
Okay. Thanks, Ever. Hello, everyone. I'm Raphaela, and I will introduce our speaker today, Francesca Bietto, with her project about development of in vitro infant intestinal models to assess responses to infant formulae. Francesca is a PhD researcher in the Food Bioavailability Lab from Teagasc Food Research Center and the University College Cork. During her doctoral research, she developed the first lab-grown model of an infant's gut, enabling scientists to study how baby formulas affect gut development. Her work aims to create safer and more effective formulas to give every child a healthier start in life. Now, let's hear from Francesca.
Hi, everyone, and thank you for joining me today. I'm very happy to share the work I've been doing as part of my PhD, the INFBAR project, which stands for Infant Intestinal Barrier. This project focuses on developing in vitro models that better represent the infant gut. Because you might think that the infant gut is a miniature version of the adult, but it's not. At birth, it's naturally more permeable, and this allows nutrients from human milk to go through and guide development. Over time, this barrier should become tighter, more selective, and more protective, and human milk guide this process, while infant formula falls short in replicating this aspect. Yet, what we use in vitro to test infant formula, it's an adult-like in vitro model, not an infant one. So my project aims at closing this gap. This background is summarized in the lit review, which is the first chapter of my thesis. In terms of the research chapters, I try to tackle the problem with different points of view. I first studied sodium butyrate to induce a leaky state in cacao monolayers. Then I evaluated sodium glycodeoxycholate as a reversible modulator of the in vitro gut barrier. In chapter 4, I compared human milk and infant formula, and I studied their effects on the newly developed infant-like model. And finally, in chapter 5, I optimized the culture conditions for the newborn-derived intestinal cell model IVEC-J2. So we started by asking ourselves a simple question. What can we use to create a leaky state using cacao cells? One candidate was sodium butyrate, a short-chain fatty acid naturally produced in the gut. At physiological concentration, sodium butyrate helps maintain epithelial integrity. However, at high doses, it can do the opposite, increasing permeability. This happens because at high concentrations, sodium butyrate can enter the cells and act as a epigenetic regulator. So my work was based on a previous study performed in my lab, in which high doses of sodium butyrate showed a decrease in tear and increase in paracellular permeability. These effects were, however, linked to the high osmolality of the sodium butyrate solution. So in the current study, I try to understand the mechanism of action and differentiate what effects were sodium butyrate specific and what effects were due to osmolality. To do so, I used an osmotic control, mannitol. Mannitol is a sugar that is not metabolized nor internalized by the cells. So I created solutions of mannitol that matched the osmolality of sodium butyrate, and I used this treatment in parallel to study tear, the metabolism of the cells, and the inflammation. To therefore check if the teardrop we saw was due to osmotic stress, I exposed for 24 hours CaCO2 monolayers to sodium butyrate at a concentration ranging from 50 to 150 millimolar, and to mannitol at a concentration from 100 to 250 millimolar. These mannitol solutions matched perfectly the osmolality of all of these sodium butyrate concentrations. So in the 24 hours treatment, we observed a drop in tear for both sodium butyrate and mannitol. But what was interesting is that upon removal of the treatment, sodium butyrate did not allow recovery, while mannitol allowed it. This recovery is particularly important when modeling an infant-like gut because we want a cell model that's able to mature into an adult one, to be able to study ingredients or formula that speed up this process. To further study the osmolality, the effect of osmolality, we created isosmotic solutions of sodium butyrate and mannitol. What does that mean? We diluted DMAM with milk water, and we resuspended the sodium butyrate and the mannitol in these media. Doing so, we obtained solutions of mannitol and sodium butyrate that had an osmolality of 360 milliols per kilo, which is the physiological isosmotic condition. With these treatments, we observed no teardrop for any of the concentrations. So we can conclude that the teardrop, it is due to osmolality. But sodium butyrate adds an extra biological component that makes the effect stronger and less reversible. Next, we wanted to know whether sodium butyrate was actually taken up by the cells and metabolized. So we measured the concentration of both sodium butyrate and mannitol in the apical at time 0 and in the apical and the basolateral after 24 hours incubation. Both compounds crossed the monolayers, but only sodium butyrate showed an internalization. In fact, the sum of the concentrations found in the apical and basolateral after 24 hours exposure was not making up 100% of what was applied to the cells at time 0. So this means that sodium butyrate was internalized. To study if it was also metabolized, we performed the MTS assay, which measures the metabolic activity of the cells. And for both solutions, both conditions, there was no increase in the MTS, therefore no metabolism of these components. When we looked at the ATP content, which is another indication of metabolic activity, we observed that sodium butyrate and mannitol both decrease the ATP content inside the cells. So they disrupted the cellular energy balance. Sodium butyrate was doing so at a higher strength compared to mannitol. To look further into the mechanism of action, we studied apoptosis and the stress markers. We chose 2 concentrations of sodium butyrate and they're matching mannitol solutions. The 2 concentrations were 75 millimolar, which was the lowest dose that led to a decrease in tear and an increase in paracellular permeability, and 125 millimolar, which was the reference dose from our previous study. We observed that both mannitol and sodium butyrate increased caspase-3 activity, therefore induced apoptosis, but only sodium butyrate was inducing the gene expression of the cell markers, cell stress markers that we studied, which were the cytokines, the heat shock protein 70, and the P21. Also, interestingly, after exposure to sodium butyrate, the cells were also exposed to dexamethasone, which is a standard steroid. Dexamethasone induced an increase in tear by 45% and a normalization of the stress markers compared to control. So this means that sodium butyrate induced a damage that was partly reversible under standard steroid treatment. So to conclude, we can say that sodium butyrate induced irreversible barrier loss with increased permeability and cellular stress. Mannitol instead was inducing a tear decrease and an increase in permeability, but this wasn't accompanied by cellular stress. Moreover, the treatment of the sodium butyrate monolayer with dexamethasone induced an increase in tear and a normalization of the stress markers. So this model, the sodium butyrate model, resembles a leaky gut that can be reversed by a steroid treatment, which is the type of inflammation that we find in the gut when we have conditions such as necrotizing enterocolitis or IBD. So the NAB model could represent an inflamed leaky gut. So as sodium butyrate couldn't be used for the healthy infant intestine, we moved on and we studied our next candidate, sodium glycodeoxycholate. Sodium glycodeoxycholate is a biosalt that has been studied in vitro and in vivo to improve oral drug delivery through increasing permeability. So we selected concentration of GDC from 0.5 to 1 millimolar. We exposed them to monolayers of CACO2 and HT29 in co-cultures for 2 hours, and we studied the permeability. First of all, we wanted to make sure that GDC was non-toxic. So we performed an NTS for viability, and we found an IC50 of 2.49 millimolar. We studied the LDH release and the caspase-3 activity and the inflammatory response. We found no inflammation and no cell death. So as we ticked these boxes, we then tested sodium glycodeoxycholate for increased permeability. And we found that after 2 hours incubation, sodium glycodeoxycholate was decreasing tear, however, allowing recovery upon removal, and induced higher lactose permeability. Particularly, 0.8 millimolar was the concentration, the lowest concentration, inducing an effect. Then we moved on into characterizing the phenotype induced by GDC. We observed that an increased acidic mucus staining with Alcian blue in the dose response. And this is similar to what happens in vivo, because in the infant, we find a mucus that's more acidic and more permeable. We also observed an increased activity of alkaline phosphatase in the apical side. This again mirrors the high alkaline phosphatase found in the infant in vivo. Finally, we observed also an altered tight junction profile with 0.8 millimolar GDC, inducing an increase in the expression of Claudin 2 and 4, JAN1, and a decrease in occludin fluorescence. This tight junction profile mimics the infant, in which we found a decreased occludin and increased Claudin 2. So 0.8 millimolar was again the lowest concentration inducing an effect, so it was chosen as the best candidate. So we studied the 0.8 millimolar concentrations in terms of cell structure. We found that 0.8 millimolar was decreasing the occludin intensity, but it was also decreasing the occludin-actin colocalization, that is measured with the Pearson's coefficient and the Manders coefficient. This decrease in occludin-actin colocalization indicates that occludin was less tightly linked to the actin cytoskeleton, reflecting a more permeable and adaptable junction structure. Interestingly, we also observed a slight decrease, but still significant, in the monolayer's thickness. This is more physiologically similar to the native intestinal epithelium compared to standard cockatoo monolayers. So with this reversible infant-like model in place, we compared human milk with three different, the three commercially available infant formula, which are dairy, soya, and amino acid. The amino acid infant formula contains no protein, and every amino acid is added individually. This is particularly suitable for those children that have allergies. So we digested these foods with the infant in vitro digestion, and we exposed our adult-like and infant-like monolayers for 2 hours to these digested foods. Our aim was to study nutrient absorption and gut barrier maturation. Regarding nutrient absorption, we use mass spectrometry based peptidomic to profile apical and basolateral peptides. This slide here shows the unique peptides detected in every compartment. What we can see is that in the basolateral compartment, the infant-like monolayers had more unique peptides compared to the adult-like. Also, what's interesting here is that the apical peptides were more abundant than the digesta peptides. This means that in the apical side of the cells, there's a further digestion of the peptides due to brush border peptides. Also, what's interesting is that these peptides that were apical only are more abundant in the infant compared to the adult. This is in line with what happens in vivo, in which the infant apical brush border enzymes are more active, like the peptidases are more active compared to the adult. To further investigate the action of brush border enzymes, we mapped the apical only peptides back to their longer digesta precursor using enzyme-specific cleavage motifs. And the resulting pattern clearly supports the activity of brush border enzymes on the cells, generating new and shorter peptides. Importantly, soya and human milk, infant soya and human milk produce more of these brush border match cleavage events compared to the adult condition. Again, supporting a higher activity of brush border enzyme in the infant-like monolayers. Next, we also looked at not only at how many peptides cross the barrier, but their abundancy. This graph here is a Venn chart that shows on the x-axis the fold change between the infant and the adult-like model for the intensity of each peptide, while the y-axis is the p-value. So we are mostly interested in the in the top right quadrant, which is the significantly higher peptides in the infant compared to the adult. Well, here in the basolateral for human milk, we can see that there are many peptides that are significantly enriched in the infant compared to the adult. Also, of these peptides, we found that 5 were bioactive, and 4 out of 5 of these bioactive peptides were more abundant in the infant compared to the adult. So this supports the idea that the infant-like model not only induces a higher transport of the peptides derived from human milk, but also increases the bioactive ones. So we also examined the tight junction structure after exposure, and we observed that in the infant-like model, when exposed to human milk, occludin intensified in certain areas, and this effect was not visible for the other infant formula or for the adult-like model. Also, the occludin-actin colocalization for human milk was higher than in the other conditions. So this shows that our infant model can mature when exposed to human milk. These effects were not parallel in the adult-like model. So all in all, we can say that our 0.8 millimolar GDC model shows a strong promise in studying nutrient absorption and gut barrier maturation. And this hold promise because with this model, we will be able to rate infant formula and improving them against the human milk-like functional benchmark. So finally, my final chapter, in parallel to the human-like cells, I also worked on optimizing a piglet-derived intestinal cell line, the IPEC-J2. The IPEC-J2 is a non-cancerous cell line derived from the jejunum of the newborn piglet, and it naturally expresses nutrient transporters and tight junctions. So it's a holds promise into being used for nutrient transport. However, it has some drawbacks. One of them is that it was never used for nutrient transport studies. And second, the tear and the paracellular transport are very variable in the literature. So I worked on optimizing and the culture conditions and the media to grow these cells for nutritional studies. I used 4 medias that are mostly present in the literature. PS10, which is DMEM supplemented with 10% porcine serum. PS5 plus EGF and ETS that are supplements that induce differentiation and proliferation. PS10 plus the WRN factor, which is a supplement used in the organoid culture for maintaining stem cells. And finally, FBS10, which is 10% FBS, that was the media recommended by the culture collection. Growing the cells in these 4 medias, I studied the monolayer characteristic, and I exposed the monolayers to digested infant milk formula. I firstly studied the transepithelial electrical resistance, the TEER, to compare different medias. And what I observed is that PS10 and PS5 both showed stable TEERs, values that were stable from day 12, and these matched the lactulose permeability. In contrast, PS10 plus WRN and FBS10 induced a tear that was not physiological, it was too high. These monolayers also had a low alkaline phosphatase and LDH activity, showing poor differentiation. Moreover, the lactulose permeability for these 2 conditions wasn't matching what observed with the tear. In fact, it was an intermediate lactulose permeability, while the tear was very high. So all in all, we can say that PS10 and PS5 plus EGF and ITS were the most promising media with a stable physiological tear and a differentiated monolayers. When we looked at the cell structure using zonal fluorescence, we saw a distinct architecture between PS10 and PS5. Specifically, PS10 had a higher zonulin and bigger cells compared to PS5 plus EGF that had lower zonulin and smaller and more abundant cells. At the gene level, the expression patterns confirm these structural differences. In PS5, we saw an increase of S100A4, which is a fibroblast-associated marker, suggesting a less differentiated and mesenchymal-like state for these media. PS10, on the other hand, show a higher expression of some nutrient transporters, showing a more functional and absorptive phenotype. PS10 plus WRN showed an increase in MUC2, consistent with a secretory differentiated phenotype. And finally, PS10 had a higher level of occludin and zonulin that are associated with the high tear and very tight monolayers. We didn't detect enteroendocrine an immune cells marker. So overall, we can say that the IPEC J2 monolayers are differentiated cells that differentiate into epithelial cells. That overall PS10 provides the most differentiated and functionally relevant epithelial profile. So we therefore chose the PS10 and the PS5 to further study these monolayers and expose them to digested infant formula. We chose two different methods of inactivation of the digesta, the heat treatment and Pefabloc plus orlistat. We performed an MTS assay to decide on the concentration to use, and we found that heat inactivation allowed the 250 microgram protein per square centimeter, while Pefabloc and orlistat only 150. When after exposure, we observed that PS10 allowed an increase in tear when with the heat inactivated infant formula. PS10 also induced a higher basolateral transport of amino acid when exposed to the IMF with Pefabloc and orlistat. This is in line with the gene expression in which PS10 induced an increase of 2 different amino acid transporters compared to PS5. So all in all, we can say that the IPEC J2 cell line represents a valuable intestinal model expressing nutrient transporters and tight junction proteins. However, the cell culture medium strongly influences the barrier integrity and permeability. PS10 is the optimal condition, producing a stable tear, increasing zonulin, high ALP and LDH, and high transporters gene expression. When exposed to infant formula, IPEC J2 monolayer in PS10 allow an increase in tear and a high basolateral transport of amino acid, showing that this monolayer is responsive to exposure to digested foods. So to bring everything together, what did I actually achieve with this project? First, I characterized sodium butyrate as a tool to create an inflamed leaky gut. Then I developed an in vitro model of the infant gut barrier, and I used it to evaluate gut maturation induced by human milk or infant formula. Finally, I developed a protocol for the newborn piglet cell line IPEC J2. But why does this matter outside the lab? Because together, these models form a toolbox that brings benefits on several levels. For society, it means improve infant nutrition and health. For industry, it means a method to optimize infant formula. And for science, it provides a new in vitro model to study nutrient absorption. So in short, this work helped connecting research, innovation, and public health, all aimed at giving every infant the healthiest possible start in life. Looking ahead, the next step is to make these models even more physiologically complete, for example, by measuring enzyme and transporters activity, adding immune cells, and eventually adding a microbiota component. This would bring us even close to a realistic infant gut model for nutrition research. Now that's all, folks. And I just wanted to give a special thanks to my supervisors, in particular to Dr. Linda Giblin from Teagasc. I also want to thank all of my collaborators, the CIAL in Spain, the CNR in Italy, and the INRAE in France. And I want to thank you for your attention. And now I'm happy to take your questions. Thank you.
So, yeah, Francesca, great talk. I would like to thank you from the side of organizational part. Thank you for your very nice, interesting research field and very important research on a very important research area, infant formula. Great. Thank you so much. And I guess it was already a perfect trial for your upcoming Viva.
It was.
Thank you.
It was.
So we have a lot of attendees, so I would like to encourage people to type your questions if you still have unanswered questions in our Q&A box. But maybe to like break the ice, I can start with the first question. So Francesca, it's a very, very like difficult topic, I guess. So if you're talking to your grandparents, can you like sum up the most important highlights of your work in a very maybe easy language just for non-specialists?
Yeah, so I would say that a newborn baby has an intestine that allows more nutrients to go through because they need those nutrients from human milk to grow and to develop. We need to have good systems to test, to develop infant formula because not everyone can or want to breastfeed. So we need to be able to create infant formula that match the effect of human milk. And to do so, I took intestinal cells, I grew them, and I tried to make them more similar to what an infant is.
Mm-hmm. Cool, perfect. So I'm completely new on this topic. This was a perfect explanation for me. Thank you. So you developed also a new model. Maybe can you compare it like the most used CACO2 model, and maybe can you like figure out some advantages of your model?
So, so first of all, when modeling an infant model, it is hard to have an in vivo benchmark because obviously we don't have many data available on in vivo children for obvious ethical reasons. So the only reliable data we have is this lactulose transport. So basically, you can for this assay, you can feed children with a solution of lactulose and mannitol that are two sugars that go through that are not used up by the human body. So they go through your system and they are excreted by the urine. So by measuring the rate of excretion, we can go back and find the permeability of the intestine. So that's the only data we have in vivo. And that in the literature, it is shown that between 3 months and 15 months of age, there's a decrease in this lactulose permeability in the intestine of around 55%. With my model, the CACO2 exposed to 0.8 millimolar GDC, I observed an increase in permeability that was around the 60%. So closely, let's say, goes close to that value. Regarding all of the other features of the intestinal cells, so the transporters, the tight junction, it is harder to compare it in vivo and in vitro because all the data we have is based on animal model, rats or pigs.
Correct.
Yeah.
Wow, thank you so much. So I'm just checking the Q&A box. There's still no questions. Maybe ever. I will.
Yeah, I can actually come with one question. So even you mentioned some like the future steps to improve the methods, to go further in your research. But I know that the ring trial is something that for modeling it's important to standardize model. Do you have any idea if at least in your group or in the INFOGEST working group on absorption, is it planning to go further like with a ring trial with your model or something like that?
Okay, well, there's not a plan yet, but I think it'd be very, very useful, especially for the IPEC J2 cell line. I've already worked very closely with Miriam Grandi in INRAE, and we worked together in parallel on these IPEC J2 cells. We observed that, growing these cells is quite hard. It's very variable. The permeability changes based on operational procedures, the FBS, the serum batch, even the transwell insert pore numbers. So I think a coordinated ring trial on that would be the next logical step to be able to use the cell line.
Okay. And I have another question. I think it was in the chapter on the IPEC cells that you use like different inactivation methods for the digesta. And I think you get like different results on the concentration of the digesta that you can expose to the cells. So maybe can you just give us what is the pros or cons and using one of the other, or well, I guess there's something that causes toxicity to the cells. Maybe just give us a bit more of that.
Yeah, so heat inactivation. So for the chapter in which I exposed the GDC model to the digesta, I chose pefablock and orlistat over heat inactivation.
Yeah.
This is because heat inactivation denaturates the proteins, and I think that was important because infant formula are heat inactivated, are heat treated for safety reasons, while human milk is not. So a difference in the two foods could rely on that step. So I decided to use pefablock and orlistat. However, pefablock and orlistat can interfere with the cell's monolayer. And this was explained very well in a recent review by Dr. Alina Kondrashina. So yeah, pefablock and orlistat could cause mild toxicity and interfere with the cells. So for the IPEC J2 cell line, as it's a new cell line, I decided to use both inactivation methods. And in fact, pefablock and orlistat was more toxic compared to the heat inactivated one. So that's why I think heat inactivated food gave an increase in TEER. I think it's because it was more concentrated. While for the amino acid transport, I had a better result with pefablock and orlistat, and I think that's because as you have the natural structure of the proteins that could influence the activity of the apical brush border enzyme and further digestion and transport of the amino acids.
Okay. Thank you so much. I think one of the attendees raised the hand, but I don't know if we can just give the chance, like maybe if someone can just can use the Q&A for the question. Okay, maybe I can just give allow to talk here. Yeah.
Can you hear me?
Yes, we can hear you, Christina.
Well, it's Raquel speaking.
Okay, sorry.
Thank you, Francesca. I think it was a great seminar. I was wondering, when you do the digestion before you treat the cells, do you separate like the bioaccessible fraction and non-accessible bioaccessible fraction? I mean, do you centrifuge the digester before treating the cells?
No, I did not. I filtered the sterile filter, but I did not separate the bioaccessible. But I did for the amino acid study. Okay,
and did you find any difficulties in separating both fractions? Like when we do it, we find like fat that is on the top of the tube. And so we are very careful to take like no fat to treat the cells. Do you do the same, or do you do something different?
No, I did not. Like I see where you're coming from, especially for the human milk. That's what happens. But I did not, because I originally did not only want to study proteins, but also like the human milk or the infant formula as a whole food. And that's why I didn't centrifuge. I only sterile filter. And because also the biological effects on the cells from deriving from human milk can be due to many other nutrients, not only peptides. It could be due to fats, it could be due to oligosaccharides. So that's why I did not, because for this initial part of the study, I wanted to study the food as a whole. Yeah.
Okay. Okay, thank you.
Okay. So I don't know if we have more questions. No. Okay. Probably I can just thank Francesca for this presentation, for the discussion and everything. We wish you like the best of the luck for your Viva. We know that you are
Thanks.
close to your final step of your PhD. So all the best. And now I'm gonna move to the announce for the next webinar. So I'm gonna share my screen. Okay, can you see my screen? Okay, so for the next webinar in the 3rd of December, we will have Marina Eichhorn from the Technische Universität Berlin. So she's actually just a recent PhD. So she presented her Viva in the last September. So she will present the findings of her PhD in December. So we invite you all to attend this webinar. And then we're looking for speakers for next year. So if you are interested on sharing your results from your PhD, even if you're a PhD student in the last stage, if you recently defended your Viva, or if you are a postdoc or young researcher, just reach us out and we can just use this space to disseminate your results. Then another announcement is, okay, this is just the whole team. Our team is actually growing. Then we are kind of excited for our conference in next year in Gdańsk in Poland. So the abstract submission is not open yet, but this is just the opportunity to say save the date, start preparing your abstracts, to start thinking about it, and that's it. So then we can say thank you so much, Raphaela, Francesca, Marina, and we see you in the next webinar in December. Okay,
Thank you
bye-bye.
so much. Bye-bye.
Bye.